my slides visible? Okay, perfect. Hello, everyone. My name is Tanvi Pathrikar. I am a PhD candidate in Ambika Vajpayee's lab at Northeastern University in the Bioengineering department. Thank you for this opportunity to present my work on Cationic Exosomes Anchored with Receptor Antagonist of Interleukin-1 for Cartilage Targeting and Repair. Osteoarthritis is a debilitating joint condition that affects joint tissues like cartilage, synovium, meniscus, and ligaments. It affects over 500 million people globally and results in severe inflammation, chronic pain, and loss of mobility and function in the joint. Although there are several factors that can trigger OA, we are focused on post-traumatic OA or PTOA due to injury or trauma to the joint. About 90% of all ACL injuries result in OA progression over a period of 8 to 10 years. The rapid progression of PTOA enables the opportunity for early intervention before the disease progresses to an irreversible state. Since OA is a localized condition, an intra-articular injection or IA injection is more efficacious than systemic administration as it prevents the side effects. However, both small and large molecules, when injected in the joint space, get cleared out rapidly by subsynovial capillaries or lymphatics, respectively. As a result, high doses or frequent injections are needed, which is not feasible. Therefore, effective delivery systems are needed to enable rapid cartilage uptake before clearance from the joint space. Cartilage tissue is a very dense, avascular, aneural, and alymphatic tissue. It is made up of a dense meshwork of collagen 2 fibers and aggregans. This is a structure of a 300 megadalton aggregan aggregate. It is made up of hundreds of 3 megadalton aggregan monomers, which contain negatively charged chondroitin and keratin sulfate GAG chains that are spaced 2 to 4 nanometers apart from each other. This matrix gives cartilage a nanopore size of about 10 nanometers. The aggregate meshwork gets denser as we go deeper in the cartilage and makes it even more difficult for solutes and molecules to penetrate through the tissue. Chondrocytes, the cells responsible for maintaining the ECM and producing the matrix components, abundantly reside in the deep zone of the cartilage tissue. Considering the rapid clearance from the joint, along with the high negative charge and complex nature of the cartilage tissue, drug delivery to cartilage is extremely difficult and challenging. Interleukin-1 or IL-1 is one of the main pro-inflammatory cytokines that is elevated in OA. It binds to the IL-1R1 receptor and activates multiple signaling pathways such as NF-kappa B and MAPK. The signal cascade results in upregulation of OA markers such as MMPs and ADAMTS, which results in catabolic reactions like proteoglycan degradation and collagen disruption. When IL-1 binds to the IL-1R1 receptor, it forms a high affinity complex with the IL-1R accessory protein. IL-1RA, the receptor antagonist of IL-1, competitively binds with the IL-1R1 receptor and prevents the recruitment of the IL-1R accessory protein and thus inhibiting the signaling pathways. As a result, IL-1RA has been considered as a DMOD or a disease-modifying osteoarthritic drug. In fact, IL-1RA is FDA approved for rheumatoid arthritis, which is a systemic disease, but has failed for OA treatment due to lack of local drug delivery and cartilage targeting approaches. Previous work from our lab evaluated the effects of single versus sustained dose of IL-1RA in an IL-1 treated cartilage explant culture model. In this first graph, this black curve represents the healthy control in which we can see a gradual increase in the GAG loss as expected. The red curve shows IL-1 or arthritic condition in which the GAG loss is significantly elevated. The blue curve represents a single dose of IL-1RA, which mimics the rapid clearance of IL-1RA from the joint and thus is unable to suppress the GAG loss. This gray curve represents a sustained dose, which means that the amount of IL-1RA was maintained at a sustained dose by continuously adding it at every media change. And thus, this sustained dose suppressed the GAG loss levels down close to control levels. Additionally, while a sustained dose was able to restore the cell metabolism and return the chondrocytes to control levels, a single dose was unable to restore the metabolic activity of the cells that had been decreased by the IL-1 treatment. Further, cartilage viability images of the control group show green fluorescence indicating majority live cells, whereas red fluorescence in the IL-1 group indicates dead cells. Single dose of IL-1RA was unable to rescue the cartilage and looks similar to the IL-1 group. However, with a sustained dose, it was successful in suppressing the IL-1-induced GAG loss and has a majority of live cells. This shows that IL-1RA is an effective drug, but a sustained dose is required. So how do we overcome these challenges for delivery of IL-1RA? Exosomes are 40 to 200 nanometer sized mini cell copies that are packed with genetic materials, growth factors, and cytokines. They have found applications in drug delivery due to their high biocompatibility and role in intercellular communication. Recent work has shown that MSC-derived exosomes have intrinsic therapeutic potential. For example, here, MSC-derived exosomes were intraarticularly injected into DMM-OA rat models 4 weeks post-surgery, and they rescued the injury-induced cartilage better than the saline control as seen in the safranin O images. Thus, our goal is to synthesize cartilage-targeting cationic exosomes that can penetrate through the full thickness of cartilage to reach chondrocytes and deliver a single sustained dose of IL-1RA. The high negative charge of cartilage provides a unique opportunity to use electrostatic interactions to enhance drug delivery systems and making them positively charged. The concentration of positively charged drug carriers at the synovial fluid cartilage interface is enhanced by a factor K known as Donan partitioning. This interface concentration creates a higher concentration gradient, which leads to faster diffusion rate along the cartilage thickness and higher uptake. However, too high of a positive charge will hinder diffusion of the carrier due to strong binding interactions. The net positive charge of the carrier has to be optimized in such a way that it enables weak reversible binding interactions for delivery to the deep zones of cartilage tissue. Our lab has designed cartilage targeting optimally charged cationic motifs such as avidin and cationic peptide carriers or CPCs, which due to their optimal size and negative charge, net charge, can penetrate through the full thickness of cartilage tissue within 4 to 6 hours and result in 200 to 400 times higher intracartilage uptake compared to their neutral counterpart. We will use these motifs for reversing the charge on the exosome surface. We use differential ultracentrifugation to isolate exosomes from bovine MSCs, and then using size exclusion chromatography, we extract high purity exosomes at fractions 8 and 9. To functionalize the cationic motifs, we first anchor DSP PEG azide lipid on the exosome surface. DSP is lipophilic and inserts itself in the lipid bilayer, while PEG being hydrophilic hangs on the outside with an azide group. Then using copper-free DBCO click chemistry, we can click any cationic motifs on the exosomes to obtain cationic exosomes. So the scheme of our formulation starts by anchoring the cationic motifs on the surface of the exosomes using click chemistry, as I mentioned in the previous slide. We then encapsulate IL-1RA inside the exosomes using sonication and anchor more IL-1RA on the surface. So our final system consists of cationic motifs on the surface of the exosomes and IL-1RA that is encapsulated as well as loaded on the surface. Exoavidin IL-1RA showed a significant change in zeta potential from negative 27 millivolts to negative 2.3 millivolts. ExoCPC plus 14 shows a charge almost close to neutral. To assess the amount of cationic motifs and IL-1RA loaded on the exosomes, we fluorescently labeled them. So IL-1RA was labeled with FITC, CPC plus 14 with Sci5, and Avidin Texas Red was labeled with Texas Red. The TEM images confirm that the surface modification do not alter the size or the morphology of these cationic exosomes. Finally, we also assessed the cytotoxicity of IL-1RA loaded cationic exosomes and found that they were safe to use as they showed 100% cell viability. To further characterize our cationic exosomes, we first fluorescently labeled the motifs like I mentioned in the previous slide. So here, avidin is labeled with Texas red. The encapsulated as well as the anchored IL-1RA is labeled with FITC, and CPC is labeled with Sci5. We anchor our cationic exosomes on magnetic beads that are coated with the CD63 antibody to confirm the conjugations using flow cytometry and confocal microscopy. So the red fluorescence here confirms the presence of avidin, green confirms IL-1RA, and the overlap confirms the presence of both avidin and IL-1RA on the exosomes. Similarly, the magenta fluorescence confirms CPC plus 14, the green again confirms IL-1RA presence, and the overlap of these confirm that both CPC plus 14 and IL-1RA are present on the surface of the exosomes. Then, using a custom chamber, we studied the 1D transport of IL-1RA loaded cationic exosomes from the superficial zone to deep zone of the cartilage tissue. The explants were treated with 0.1 mg per ml of trypsin EDTA for 4 hours that resulted in about a 40% gag loss. Native exosomes that were labeled with Sci5 and loaded with IL-1RA FITC did not penetrate through the full thickness of the cartilage tissue even after 24 hours of absorption. Avidin and IL-1RA functionalized exosomes, where avidin was labeled with Texas red and IL-1RA with FITC, showed slightly better transport. However, it also was not able to penetrate through the full thickness of the cartilage. Lastly, ExoCPC plus 14 was able to penetrate through the full thickness of the cartilage tissue after 24 hours of absorption. We also evaluated the uptake in chondrocytes residing within full thickness of healthy and IL-1 treated cartilage explants with about a 35% gag loss. We stained the nucleus blue and the chondrocyte membrane with red fluorescence. The overlapped images confirm that cationic exosomes were mostly uptaken by chondrocytes present in the IL-1 treated cartilage. Finally, we tested the systems in an in vitro model. We started by harvesting 3 mm by 1 mm cartilage explants from the femoral groove of a young bovine knee joint. These explants were then cultured in a 96-well plate. After the 48-hour free swelling period, we introduced the IL-1 treatment for another 2 days. After 2 days, we added our treatments that are shown here in this table. At the end of the media was changed every 2 days, and at the end of the experiment, which is day 8, the explants were digested. The collected media and digested explants were then used for a cumulative gag loss analysis. At the end of the experiment, we also sectioned some cartilage explants to study the viability. As we see here, the healthy control shows a gradual and uniform increase in gag loss as is expected, and is also seen in the cartilage viability images, which predominantly shows a green fluorescence indicating a majority of live cells. With the addition of IL-1 treatment, the gag loss is significantly elevated, and the dominant fluorescence that is observed is red, which indicates dead cells. Single dose of free IL-1RA was unable to suppress the gag loss, as seen in both the percent cumulative gag loss graph as well as the cartilage viability image. Similarly, exo-avidin IL-1RA was also unable to rescue the cartilage from IL-1-induced gag loss, perhaps owing to the poor transport property, as I showed earlier. However, ExoCPC plus 14 IL-1RA was able to bring the gag loss levels down close to control levels. In fact, on day 6 and day 8, there is no significant difference between the control group and ExoCPC plus 14 IL-1RA. To summarize, we for the first time engineered cationic exosomes using click chemistry to anchor cationic motifs on exosome surface to neutralize their net charge. These charge-reversed cationic exosomes showed full thickness penetration and chondrocyte targeting. When we tested the cationic exosomes in an in vitro arthritic model, they successfully delivered loaded IL-1RA to the chondrocyte and rescued IL-1 induced catabolism with only a single dose. Future studies for this project include